Hey everyone, welcome back to Windows Seed Studies. So we have been learning about histone modifications, in which we have already learned about histone acetylation as well as histone methylation. So today we are going to learn about a new histone modification, and that is histone phosphorylation. So as the name suggests, histone phosphorylation means addition of phosphate group on the histone. So this phosphate group is overall negatively charged. So histones are positively charged proteins. Why? Because histones have the basic amino acids such as lysine and arginine, which gives it an overall positive charge. Okay. So around this positively charged proteins, the DNA, which is negatively charged, is wrapped up. Okay. So but due to addition of phosphate group on the histone DNA interaction gets weak. Okay, and this is how histone phosphorylation changes the chromatin environment. So we can say that out of the two modes of functionings of histone modification, histone phosphorylation works through direct perturbation of DNA histone interactions or histone DNA interactions. Why? Because a charged moiety is being added on histone, so that is going to change histone conformation, and that will change. That will have direct effect on the histone and DNA interactions. Unlike histone methylation, which is a signal for recruitment of some factors which are going to change the chromatin environment. So that is an indirect way of changing chromatin environment, while this is the direct way of changing the chromatin environment. So it brings out the direct perturbation in the histone DNA interactions. Okay, so this is just like acetylation. In acetylation, the positive charge on histone is neutralized, but in histone phosphorylation, the negative group, the phosphate group is being added on the histones. Okay, but unlike histone acetylation or histone methylation, the histone phosphorylation is done on serine, threonine, or tyrosine residues. Okay, so this again, there's an interesting fact about histone phosphorylation that the phosphate group there are phospho-acceptor sites on all of the core histones. Like we have four core histones, right? And all of them are present twice in the nucleosome. So those are H3, H4, H2A, and H2B. So all of those histones have the phospho-acceptor site. On the H3, we have histane as well as S28. On the H4 we have S1. On H2A we have S1, and on H2B we have S14 as well as S32. Okay, so there are different enzymes which are going to do the which are going to add phosphate group on the different serine or threonine residues. So in the so we there the most studied phosphorylation mark is H3S10 pH. So this is written in this way, pH, or sometimes it is also written as P. So H3S10 pH is the most studied phosphorylation mark. So phosphorylation event is very important because it is genome wide event. Okay, so let's first take a look at the mechanism, like does phosphorylation do. So let's have a look at the consequences of phosphorylation in the cell. Okay, so what happens is that phosphorylation is going to make the DNA histone interactions weak, right? So keep this in mind. Okay, so why phosphorylation is a very interesting event, because it is very context dependent. Let's see how it is. So at the start of the prophase, like in the cell cycle, when there is when there is division stage, so at the start of prophase, phosphorylation of phosphorylation of histones is seen. Okay, so the phosphorylation of a phosphorylation event starts at the start of the prophase. But as we go in the metaphase stage, the phosphorylation event is at its peak. Okay, and as we progress to anaphase or telophase, you can see you can see that phosphorylation event is reducing, and there would be dephosphorylation. Okay. So all of you are aware of all of you are aware of the chromatin state in the cell cycle, right? So I hope you all remember that when we were learning about chromatin, we learned that the chromatin is present in the interphase stage. Okay, while in the metaphase stage, or say that in the cell division stage, we have chromosome. Okay, and chromosomes are nothing but the highly condensed form of the chromatin. So they are the high level of order. Like the packaging is at its peak in the chromosomes. Okay, so here I think that you can easily correlate. Now I guess you can easily correlate the phosphorylation to the chromatin state. So phosphorylation is important. Why? Because it is involved in the chromatin condensation. This this sounds a bit contradictory, right? So like our basic knowledge about the chromatin environment is that, see, whenever the DNA histone interactions, okay, when we were learning about epigenetic marks, we learned that whenever DNA histone interaction, they increase, or they get strong. At that time, this will condense the DNA. Condense the chromatin. Means this will pack the chromatin in. This will densely pack the chromatin. Okay, it will make it dense. And whenever DNA histone interaction goes down, like they gets weak, so this is going to loosen up the DNA, right? Because if the histones and DNA interactions are not strong enough to have that DNA tightly wrapped up around histones, right? So whenever they are weak, it is going to make the chromatin loose. Okay, that this is what we have learned till now. But here you now I already told you that the phosphorylation is going to make the DNA histone interaction weak. So logically, it should lose the chromatin and it should work as an active mark. So but it should release the packing. Okay, it shouldn't make the chromatin dense. But here you can see at the metaphase you need chromosome structure. Okay, and it is highly packed structure, so there should be no or less phosphorylation, but we observed that the phosphorylation is at its peak during metaphase. So this is a bit weird, right? So that's it. That is why I told you that phosphorylation is very interesting. So what happens is that phosphorylation makes the DNA histone interaction weak, but what happens is that due to weakening of DNA histone interaction, okay, at the metaphase, okay, not at any cell stage, but only at metaphase. And how? Not by acetylation, only by phosphorylation. So during acetylation, the event doesn't happen. This happens only when phosphorylation is done. So during phosphorylation, it weakens the DNA histone interaction at metaphase, and this promotes the polyamines binding. Polyamine binding. So where does it bind? It binds to the DNA. Okay. So what are polyamines? So polyamines are negatively charged moieties. So they are positively charged. Okay, and they are very small. So compared to histone, they bind more extensively to DNA. Okay. Okay. So what happens is that first, so assume that is the this is your nucleosome. Okay, the octamer, sorry, and around which this is the DNA. It is wrapped around those histones. So what happens is that this when it becomes loose, the polyamines come and they bind here. Okay, they bind to DNA. Okay, so they do not bind to here, they bind to the DNA. Okay, so this is how they bind to DNA. I'm drawing a rough schematic for you to understand. So, have you ever wondered, like, that wrapping the DNA around histone makes it tightly packed, right? But those histones are positively charged. Okay, those are positively charged. So overall, the octamer is going to have a positive charge. So there will be a repulsion between those positively charged nucleosomes, right? Then how it is tightly packed in cells? As you this solves the positive charge on the histone solves the problem of DNA binding, like the positive and negative charge gets attracted, but the two nucleosomes which has overall positive charge, they are repulsed, right? So this repulsion is minimized by polyamines. Okay, so polyamines play a role. So those are also positively charged moieties, but they go bind to the DNA, and the binding the binding capacity of polyamines is more extensive than the histone. So now this polyamine is positively charged and it is bind to this DNA, and it will also have some attraction force to the DNA, which is wrapped up around on the next nucleosome. So this is going to so polyamine binding going to make or it is going to reduce nucleosomal repulsions. Okay, so now tell me what will happen if the repulsion between those nucleosomes is reduced due to polyamines, which are like they are acting like a middleman. Okay, so they are interacting with the DNA on the both nucleosomes. So they are a kind of a middleman which have like imagine they are sitting here. Okay, and they have two hands, so they are stretching like this is just imaginary. Okay, you have to imagine it. This doesn't happen literally. So they are like a middleman who has two hands and who is pulling together, who is not letting the two nucleosome, which has the opposite charge, go in other direction. So this polyamines, they are going to reduce the nucleosomal repulsion, and this reduction in nucleosomal repulsion, it enhances the packing. So due to this, the chromatin becomes dense. Okay. So I hope now you got it, like how the weakening of DNA histone interaction actually results into the dense packaging. So not this weakening of histone DNA interaction doesn't do it directly, but it induces or it promotes the polyamine binding, which is eventually going to make the chromatin more dense. But this doesn't happen during acetylation. Okay, but why? Because during acetylation, it only loosens up the DNA and it makes DNA more accessible at the regions which are going to be which are going to need, and they are going to be actively transcribing. Okay, but at the phosphorylation, which is going to happen at metaphase, it weakens up the DNA histone interaction, and then it increases the polyamine binding, which makes the DNA more condensed. So that is why during prophase, the phosphorylation starts at the metaphase. It goes at its peak, and at that point of time, you will see the chromosome highly packed structure. And then during anaphase and telophase, you will see dephosphorylation, which is going to restore the original chromatin form. Okay. So this was how the DNA phosphorylation works. Sorry, histone phosphorylation works. So that is why I told you that at the start that histone phosphorylation is more into like it is context dependent. Why? Because at the interface, it is going to loosen up the chromatin. Why? Because it is weakening the DNA histone interactions, right? So this will make the chromatin loose. But what happens at the metaphase is this makes the DNA histone interaction weak, but this making it weak promotes polyamine binding to DNA, which makes chromatin dense and converts it into the chromosome. Okay, so it is very context dependent. If you are talking about the phosphorylation at interface, it is going to loosen up the chromatin and it will make DNA more accessible. Okay, but when you talk it about in the metaphase context, it is going to make the chromatin highly dense, and it will result in chromosome formation. So that is why I said that the phosphorylation is context dependent. Okay, so I hope you will always remember this point. So yeah, this is how the phosphorylation mechanism works. Let's now take a look at the enzymes that are involved in the phosphorylation. So I told you that all core histones have the sites for phosphorylation. So H3 has S10 as well as S28. H4 has S1. H2A also has S1, and H2B has S14 as well as S32. So H3, the most studied one is H3 S10 phosphorylation. So the S10 phosphorylation is done by various enzymes in various contexts. Okay, so the enzymes which are going to phosphorylate the histone, they are known as kinases. And I hope in the cell biology or in other in other courses you already have learnt about kinase. So kinases are the enzymes which are going to phosphorylate their substrate. Okay, so histone kinases will phosphorylate histone. So histone kinases are very a specific category of enzyme. Why? Because in unlike histone methylases or histone acetylases, which only work on histone, those histone kinases have histones as their substrates, as well as there are other proteins which they can phosphorylate. So this is a distinct characteristic of kinases. Like they can phosphorylate not only histones but other proteins as well. But this doesn't happen in histone methyl transferases like HKMTs or HATs. They don't show that kind of activity. So the kinases are the enzymes which are going to phosphorylate the histones, and phosphatases, they will remove the phosphate group. So they will do the dephosphorylation. Okay, so the estine, on the estine, phosphorylation can be done by various enzymes in the various contexts. So some of those are RSK2, okay, ribosomal S6 kinase 2. The other examples are MSK1 or 2, MSK1 or MSK2. Then the other example is aurora kinases. Okay, so those aurora kinases also do the S28 phosphorylation. So aurora kinase does the both S10 as well as S28 phosphorylation. The S14 phosphorylation on H2B, it is done by the MST1 kinases. Okay, you don't really need to learn all of the names of all of those kinases, but if you can, then you should. So those are the different those are the different enzymes that are going to do there on the threonine. Okay, on the threonine, the 1/9th residue of threonine, this doesn't lie on the histone tail, but this lies on the terminal of the histone. Okay, so this was, I guess, discovered in Drosophila first. So this 1/19th threonine residue, this also gets phosphorylated. So we can write it as T1. Okay, sorry, T. So we can write it as T119 pH. Okay, and it is this threonine. This is of the protein H2A. So the H2A threonine, or the 1/9th threonine residue, this get phosphorylated, and this is involved. This is in the at the C terminal. That is why this is very important one, because generally on the C terminal, histone modifications are not done. So this is very nice example. So alongside serine and threonine, there are tyrosine residues on which phosphorylation can also be done, but the serine threonine kinases are more common than the tyrosine kinases, and that is why we see tyrosine phosphorylation in much less context, but it is very important phosphorylation event, because it is involved in maintaining the cell cycle. So the cell cycle progression from the prophase to metaphase and then to anaphase. This is very important because it is somehow maintained by the tyrosine phosphorylation and all. There is a there's an important example of the phosphorylation, and that is H2AX. So this H2AX is the variant of the H2A protein. So this variant has the serine residue at the 139th position, and this is phosphorylated, and this is done during the DNA damage repair. So you can say that this is a marker of DNA damage repair. So whenever there is DNA damage repair, this 139th residue of the H2AX protein is phosphorylated. Okay, so yeah, this is it about kinases. So let's learn about phosphatases. Okay, so phosphatases are the enzymes which are going to remove the phosphate group. So they are going to do the dephosphorylation of the histones. So there are two different types of phosphatases. One are cysteine dependent phosphatases, and oh, other are metallophosphatase. Okay, so the cysteine dependent phosphatases are those are metal independent. Well, the metallophosphatases, as the name suggests, they are metal dependent. Like they require metal ion for their functioning. Okay, those cysteine dependent phosphatases are tyrosine phosphatases. They are rare, but they are very important. Okay, so they are in the context of tyrosine. Well, those metallophosphatases are mostly serine threonine specific phosphatases. Okay. So this is how phosphorylation is very important because it is context dependent, and it is involved in the cell cycle progression. So this covers all of the important points that we need to learn about histone phosphorylation. So I'll end the lecture here, and in next lecture we are going to learn about the histone ubiquitination. So till then, see you. Bye bye.